right, everyone, welcome back. We are about to get to chapter 17, and this is exciting because now we're actually going to get into some metabolic reactions, metabolic cycles and pathways. And this is pretty cool because, we've learned about several different biomolecules. We learned about carbohydrates and proteins and lipids, et cetera, et cetera. But now we're going to start seeing how some of these biomolecules can actually be utilized to drive our biological functions. So you're going to see a lot of familiar stuff. I mean, of course, you guys have probably been talking about glycolysis in some form or fashion since high school. It's certainly part of the big three, I guess you could call metabolic pathways that people typically talk about. Those being glycolysis, citric acid cycle, and electron transport, that generates the ATP from a singular molecule of glucose. So some of this will seem familiar, but also we're going to be taking a deeper look at it really in terms of the actual reaction, sort of structurally, what is happening during each step of glycolysis, looking at it from more of a chemical point of view. I want to go ahead and just tell you this up front. Make a note of this. You will need to know all of these reactions. You'll need to know the structures of the compounds, the products, the reactants, and also the enzymes that catalyze them. Without a good working knowledge of that, then it's a lot harder to understand glycolysis as a whole. So, however, and it seems pretty daunting, but really if you understand the reactions that are going on, then it becomes a lot easier to visualize what the structure should be. And a lot of times the enzyme names give you hints as well. So we'll talk a little bit about that as we go along. Okay, so glycolysis, it is, like I said, the first stage of glucose metabolism. Right? This is how we get energy. This is the start of getting energy from a singular molecule of glucose. As far as just individual energy, specific, so as far as just direct energy production, air quotes, you don't get a whole lot from glycolysis, right? It only yields 2 ATP, which you guys know are the, from biology classes, are the basically what's referred to as sort of the energy molecules that are produced. And you end up with a lot more than that. But what it also does, it produces precursors that are then shuttled into the citric acid cycle, which makes some more ATP using those precursors. And if you ever really did a fully in-depth study of biochemistry, like say hypothetically you had a second semester of this, you really get to see how all these pathways are intertwined and feed into each other. It's really a really cool, it's kind of a humbling look because it's so complex, but it's actually a really, really neat system as well. So we're going to take a look at a part of it in this chapter. All right. Perhaps even more important than the 2 ATP that are made is the other target compound that's made here as a result of glycolysis, and that's pyruvate. You also produce 2 molecules of pyruvate, and this is what you're really using as the investment to pump into the citric acid cycle. Just a quick word before I get into glycolysis as a whole, just talking about pyruvate. Yeah, this is basically, there's a couple of different net reactions you could write for glycolysis. Here's the most simplistic one. 1 molecule of glucose yields 2 pyruvates. Right? Now, you could also start including the phosphate containing things in here. So a second way to write it, and I'm going to be lazy and just continue on this one, you could say glucose plus 2 ADP, adenosine diphosphates, plus 2 free phosphates or inorganic phosphates, yield 2 pyruvates plus 2 ATP, right? So, you can write this in terms of everything, or you could just write it in terms of the precursors. I want to focus on the precursors here for just a second, but to kind of show you that there's different things that can happen depending on not only the type of organism that you are, but also the nature of the metabolic pathway and where you're located. All right? So pyruvates, they can actually have multiple fates. Some of this pyruvate can go to the citric acid cycle, and I'm going to just abbreviate it as CAC. All right? It does this whenever these pyruvates are being used aerobically. Shouldn't have drawn that other parenthesis. There we go. Yeah. So citric acid cycle is an aerobic process. It uses oxygen. And you typically see this in animals and plants. All right. Sometimes these are shuttled, depending on what else is going on with your pathways, what feedback mechanisms happen to be in place, and what your levels are. Sometimes these can be shuttled into something called anaerobic glycolysis. All right, so of course, anaerobic means that you don't utilize oxygen. So basically what happens here is that these pyruvates, let's say you don't need any more ATP, you don't need any more aerobic production or aerobic breakdown of pyruvate. Sometimes they're shuttled to the muscles. And this is where you actually find this process, specifically in contracting muscles. And eventually what happens is these get converted into lactate. Lactate is the deprotonated version of lactic acid. So another word for this, really, no, never mind. I'm not going to go there. I'm going to confuse you if I do that. So forget that last 5 seconds there. But it does produce lactate. So, for example, if you have been really overworking your muscles and everything, sometimes some pyruvates will get shuttled to the muscles, be converted into lactate. It gives you a quick little energy source there directly in your muscles, but also produces lactate/lactic acid, which we know can help make your muscles feel sore. And then, in certain types of organisms, you can have pyruvate being used in alcoholic fermentation. And this happens with yeast. So it's the classic thing. I remember when I was a kid, my dad was very much a country boy, and a lot of country stuff, but one of the things he was really good at was making his own wine, sort of redneck wine, so to speak. And so he would get cut up a bunch of potatoes and put some other kind of starchy, heavy stuff in a big glass bottle with a narrow neck, put some yeast in it, put some grapes in there, whatever else he did to flavor it up, and you put a balloon on it, and you let it sit for a certain period of time, weeks or so. And the balloon is there to collect gases that are given off, specifically carbon dioxide, during the alcoholic fermentation process. And so what you're actually doing, the starch-containing stuff within that mixture, starch is composed of glucose units. These glucose units are broken down from the starch, and the glucose goes through glycolysis and then alcoholic fermentation within the yeast. And so one of the products of alcoholic fermentation is ethanol, and that's the basis for alcoholic beverages. So kind of neat here. We're going to focus primarily on this part of it right here, the preparation of pyruvate for entering the citric acid cycle. We might touch on the other ones a little bit along the way. All right. So let's get into it. Okay. So this is basically, in a large part, there's one thing that you'll see a lot with these reactions, as well as if we get into citric acid cycle, is you'll see there's a number of redox reactions that occur. You will frequently see a carbon becoming either more electron rich or more electron poor, right? And we've talked about this in organic chemistry. I'll highlight it when that's the case. We'll also see some other certain types of processes. You know, you'll see some words that hopefully you'll remember from organic chemistry, like isomer, right? If you don't remember what isomers are, you know, maybe pause this and take a look. I'll remind you once we get to it. And there'll be a couple that we honestly probably didn't get to in OChem that I'll highlight as well. All right, well, let's just kind of start off from the beginning here. Oh, by the way, I feel sad and old by looking at this slide. It says, first of all, the picture I used here was so old that it's not showing up. You see QuickTime and a TIFF decompressor needed to see the picture. So, if you're a fan of South Park, there used to be on one of the earlier episodes a long time ago something called the Underpants Gnomes. So, look it up, Underpants Gnomes, South Park. And so it was funny. They had a little thing where they were stealing underpants from people in the middle of the night, and their thing was that it was going to make them a lot of money. So, but they didn't really know how. So it was phase 1, collect underpants. Phase 2, question marks. Phase 3, profit. So literally this slide is just a reference to that, so you can ignore it otherwise. It's a funny episode though, so you should watch it. All right, so this is going to be a series of organic reactions. The overall net, like I drew earlier, is basically that you're going to be taking glucose, you're going to use 2 ADP and 2 inorganic phosphates to produce 2 ATP and 2 pyruvates. Remember, you need to keep track of the structures and reactions that are happening along the way. Count your carbons, right? If you're missing a carbon, go back along the way, see where you lost one, because along the way, up until the halfway point of glycolysis, you should have 6 carbons. We'll eventually get to a point where those are split, and you'll have 3 carbons per reaction to keep up with. All right? So be sure to count your carbons. There's overall 10 steps. 10 steps in glycolysis divided up into 2 phases. And again, hopefully you have the PowerPoint brought up on something on a computer. But this shows a summary of those reactions. And quite honestly, this looks kind of intimidating, doesn't it? Yeah, there's a whole lot of shit going on here. But again, it's really not that bad if you understand what's going on. So what we're going to do is we're just going to sort of walk through these and see what happens in each step, and I'll highlight certain tidbits that you need to be aware of. You can see it's kind of sort of got a weird structure here, this little flow chart. It's 10 reactions, it's 2 phases. The first phase is really defined as taking that initial glucose, phosphorylating it, and then converting it to something called glyceraldehyde 3-phosphate. All right? So once you produce glyceraldehyde 3-phosphate, that's the end of phase 1. And phase 2 is all about converting that glyceraldehyde 3-phosphate into your ATP and pyruvates. All right? You'll notice that in this little picture, there's a point where the chart kind of splits into 2 legs, right? That's going to be at the end of phase 1. And that's because you actually produce 2 different molecules of glyceraldehyde 3-phosphate, and they each undergo this particular set of reactions. So that's why this has legs. If you look closely at the 2 legs here, at the reactions coming out of glyceraldehyde 3-phosphate, you'll see they're all the same, right? So if you can remember them for one side, you can remember them for the other side. That makes it a little easier. All right. So let's start at the beginning here. So you got your glucose, right? And we've got the cyclic form of glucose here. That's going to be basically our starting point. If you remember, glucose is a hemiacetal. You see the numbers here, the 1 and the 6, that tells you the carbon number, right? So if you remember with monosaccharides, we always start numbering at the anomeric carbon, right? And in glucose, carbon number 1's anomeric. And in this case, you're going to go clockwise, and that CH2OH right there at the top, that is going to be your carbon number 6, all right? So what we're going to do here is we're going to phosphorylate it, and that means we're going to take a phosphate from ATP, we're just going to snip off a phosphate group, and we're going to attach it to the OH on carbon number 6, all right? And this is going to make a phosphoric acid ester, all right? This is catalyzed by an enzyme called hexokinase, and we use magnesium plus 2 here as a cofactor to sort of help the reaction along. All right. All right, couple of things here. First, let me talk about actually, no, rewind that. First, let me talk about energy. All right. For any of these metabolic processes, in order to get the reaction to go, you've got to have a favorable energy change, right? And we learned about that in chapter 1, all the way back in chapter 1, right? You know that to be what we consider spontaneous or a favorable reaction energetically, you need to have a negative free energy change, a negative delta G, right? Some of these organic reactions are already like that. They're already what we call exergonic, right? Some of them aren't. So in some cases, we have to have a little bit of help. So you see ATP there. ATP actually serves 2 purposes here. Number 1, it serves as the phosphate source, right? We want to put a phosphate group on carbon number 6, so that's a good source. But why does it have to be ATP? Why not just use an inorganic phosphate? Why not just use a randomly floating phosphate in your body? You got plenty of them, right? So you could do that. The problem is, if you look at strictly the reaction between a phosphate and glucose to make glucose 6-phosphate, and again, this shows up better. For some reason, my iPad does not like some of the symbols here, so I apologize. But basically what happens is, if you just took a phosphate and stuck it on glucose to make the product here, that has a delta G that is positive, right? By the way, I kind of regret doing this all those years ago when I put this together, but again, I was kind of going by the book. You'll see a couple of different delta G's represented. You'll see delta G not prime. That's the delta G with the little 0 and the apostrophe, the little prime symbol, superscripted. That refers, that refers to the delta G of this reaction under what we call standard conditions. And you've talked about standard conditions in Gen Chem, right? 1 atmosphere, 25 degrees Celsius, right? You'll also see just a delta G without the prime. That refers to the actual delta G under biochemical conditions, right? So for example, standard conditions at standard temperature, you have a temperature of 25 degrees Celsius. Our bodies are not standard temperature, right? They're not 25 degrees. They're going to be warmer than that, right? So you'll see a couple of delta G's indicated here, and I'll try to make a differentiation around them whenever possible. If I want to skip ahead for just a second, you can see them in this chart as well. Honestly, probably the most practical thing to look at is going to be the delta G on the far right, that's going to really more approximate what you have under traditional biochemical conditions and not standard conditions. But anyway, bottom line, phosphorylation of glucose is not favorable. It's endergonic. It requires an input of energy, right? So if you just try to do that by itself, it's not going to work. However, if you cleave a phosphate group off of ATP, that is a favorable process, right? That releases a lot of energy. At standard conditions, you get an energy release of 30.5 kilojoules per mole. So you may remember in a previous chapter, I talked about, in some cases, you might have biochemical reactions that are energetically unfavorable. We say they're coupled to ones that are favorable. And this coupling can produce an overall negative delta G. And that's what we have here. Phosphorylation of glucose, not favorable. Cleavage of phosphate from ATP is favorable. You add them together and you get an overall net delta G. So that's why you have to have both of those processes there in the same step. The other thing I want to talk about is hexokinase. And again, I mentioned that with the enzyme names, a lot of times you can kind of get a hint of what's going on in the reaction. Here's a good example. All right, kinase. Let's take the suffix kinase. Whenever you see the term kinase, that means an enzyme is going to be taking a phosphate from ATP and putting it on something else, on some other substrate, right? The hexo in hexokinase refers to the fact that this works on hexoses. And we learned back in the last chapter that glucose is a hexose. This also could be an enzyme that could catalyze other hexoses, for instance, like galactose, that would work as well. But in this case, it's glucose. So, hexokinase takes a phosphate off ATP, sticks it on a hexose. Right? Hexokinase actually, you can see some allosteric effects with hexokinase. First of all, there's a neat little picture here that sort of shows what happens whenever a glucose binds to it. It's almost like you can think of like a little baby having its mouth open to receive food, and the spoon goes in and then it closes its mouth. You kind of see that happening here. So as a glucose enters the binding site, literally you get a conformation shift in hexokinase that sort of causes the area around the active site to close, to sort of clamp down, to better bind your glucose. You also see, like I said, allosteric or feedback effects. Excuse me, not really allosteric, but feedback effects. Hexokinase undergoes feedback inhibition. It is inhibited by glucose 6-phosphate, which is the product here, right? So, if you end up with a buildup of glucose 6-phosphate, the product of this reaction, it will in turn bind to your hexokinase and basically inhibit it. So, one of many examples of feedback mechanisms that we're going to see. All right, step 2. All right, so step 2, at the end of step 1, we have glucose 6-phosphate. Step 2 is going to be taking glucose 6-phosphate and isomerizing it into fructose 6-phosphate. So again, you get kind of a hint here from the name, right? Isomerization. What is an isomerization? Well, if you remember from organic chemistry, constitutional isomers, they're going to be two different compounds that have the same number of atoms of each type, right? So in this case, you're going to have the exact same number of carbons, hydrogens, etc., but they're linked together differently in a different, order or pattern, right? And so that's basically what happens here. The glucose 6-phosphate doesn't lose anything or gain anything as far as atoms. It just sort of rearranges. All right? So you can see, the reaction here. I'm actually going to redraw this, so bear with me. Might take a minute. I'm actually going to redraw this over here on the whiteboard because I want to show you one or two things. So let me go ahead and draw. So all right, so there's your glucose 6-phosphate. It's going to isomerize. Wow, that's a terrible pentagon. Terrible, terrible, terrible. So embarrassing. All right, 1 second. Ah. Okay. So our reactant here is glucose 6-phosphate. Our product is fructose 6-phosphate. Let me highlight a couple of things here. Number one, it's important to keep track of the carbons. Remember, the anomeric carbon over here in glucose 6-phosphate is going to be carbon number 1. So this is the carbon number 1 right over here. All right. As you can see here, basically when you isomerize and rearrange, the ring gets smaller, right? So what actually happens is this this carbon that was the anomeric carbon, it moves out of the ring now. So now this is carbon number 1 right here. And it's going to be 2, 3, 4, 5, 6, like that. Okay. Over here, it's 1, 2, 3, 4, 5, 6. All right. So that anomeric carbon shifts out, and then you can see, if you look at carbon number 1 in both cases, that carbon, as you isomerize here, it gets reduced. This is a redox reaction, right? Carbon number 1 gets reduced. One of the definitions of reduction is that you gain hydrogens, right? Well, this carbon on the left in glyceraldehyde 3-phosphate, glucose 6-phosphate has 1 hydrogen over here. Now it's got 2. All right, so that anomeric carbon gets kicked out of the ring and reduced. And also, you end up converting what used to be a hemiacetal into an acetal. Excuse me, gosh, had a brain fart. That's not a hemiacetal. Or excuse me, that's not an acetal. That is a hemiketal. My bad. Yeah. So that's basically what you're doing there. You'll notice that this one has equilibrium arrows, whereas the other one did not. So remember, look back up here. Yeah. In step 1, that reaction went to completion. You didn't go backwards at all. But step 2, have equilibrium errors. That's because the free energy change here is very small. The free energy change for isomerization, the fructose 6-phosphate, is, depending on whether you're talking about standard conditions or biological conditions, either just below 1, just below 0 or just above 0. It's very close, right? And so when you have free energy changes that are very, very close to 0, those are the ones that tend to be reversible. Step 1, at standard conditions, at standard conditions it was negative 16.7 kilojoules per mole. At biochemical conditions it was negative 33.9. Those are much larger values, so there wasn't any chance of reversing that. All right. The enzyme for this one is called glucose phosphate isomerase. And that really kind of tells the story, right? It tells you, number one, an isomerase is going to catalyze a rearrangement from one isomer to another, and the glucose phosphate part of this basically tells you that specifically this targets a glucose phosphate, a glucose with a phosphate on it, right? You'll see a lot of these enzymes get very, very specific, and sometimes they literally will only catalyze one specific reaction, and in other cases they can catalyze a wide range of them. So it's kind of interesting. All right, step 3. Step 3 is what we like to call the point of no return for these precursors, these intermediates, to go through and finish glycolysis. Glucose can go through glycolysis, but it can go through other metabolic reactions as well. Glucose 6-phosphate, it can interact with, it can continue to go through glycolysis, but it can enter other metabolic pathways as well. Fructose 6-phosphate, same thing. It can go through glycolysis to completion, or it can shuttle out and be an intermediate in another pathway. The product of step 3 is going to be what we call fructose 1,6-bisphosphate. The only thing it does is go through glycolysis. So this is our commitment point, where everything here on out really is going to go through glycolysis. Don't have any chance of shuttling it out to another pathway. All right, so in this one, you're converting fructose 6-phosphate into fructose 1,6-bisphosphate. Think about that name, bisphosphate. You're basically adding the prefix bi to the phosphate, or bis, I guess you could say. Bis and bi both mean 2, right? So fructose 1,6-bisphosphate now has 2 phosphate groups, and you can probably guess where they are. They're going to be on carbons 1 and 6, right? And you can see that here. Carbon number 6 already has one from the last reaction. Now you're going to be adding a phosphate group to carbon number 1. And where are we getting that phosphate group from? Is it from a free-floating inorganic phosphate? No. It is from ATP, just like we saw before. All right. Much like the first step, this is going to be a coupled reaction. All right. Production of fructose 1,6-bisphosphate is endergonic, but by coupling it to exergonic cleavage of ATP, you get overall a net delta G that's very positive. That's very favorable. It's very negative. You'll see the enzyme here is called phosphofructokinase. Again, kinases, what do? They snip phosphate groups off ATP and put them on something else. The phosphofructo here basically tells you that this works on phosphorylated fructose molecules, which we have. All right. Phosphofructokinase is kind of interesting. Couple of things about it. Get back to my whiteboard. There we go. Phosphofructokinase is an example of something called an isozyme. All right. Isozymes are basically, where you can have slightly different forms of essentially the same enzyme, right? So phosphofructokinase Its composition can vary slightly depending on where it's located. It's composed basically of 4 different subunits. All right, and the book does a good job of simplistically representing those. The 2 subunits are either going to be called, 2 types of subunits are going to be called M and L, right? And this basically stands for, the M stands for muscle, and the L stands for liver, right? And there's 4 units composed of these 2 types. So, if we're looking at 4 subunits, you could have 4 M's together. You could have 4 L's together. And you could have anything in between. I think you get the idea, but for some reason I'm compelled to keep drawing these because it feels incomplete if I don't. So please forget my obsessive compulsiveness here. That's a 3. All right. So the composition will change depending on where it's located. These M4s tend to be in the muscles. L4s tend to be in the liver. And the various combinations thereof tend to be in the red blood cells. But they all do serve basically the purpose of catalyzing reactions such as this. All right, phosphofructokinase is also an enzyme that demonstrates allosteric effects. Okay. Did I write this in here? No. Okay. Let me go back to the whiteboard. There are certain things that inhibit phosphofructokinase. It's going to be inhibited by ATP. So if you have an excess of ATP, it'll loop back, bind with this enzyme, and make it less active. It's inhibited by fructose 1,6 bisphosphate, which is the product of this reaction. And it's also inhibited by another isomer of this, fructose 2,6 bisphosphate. So all these act to inhibit. If you're to look at an enzyme kinetics plot for phosphofructokinase, it's going to look awfully familiar. Well, again, on my iPad it looks terrible, but if you look at it on a computer, you'll see, just like we saw previously in enzyme chapters, you can have different levels of you have certain conditions where it can look more hyperbolic of a shape and certain conditions where it looks more sigmoidal, right? You really see these allosteric effects kicked in whenever you have some, in this case they use ATP as an example, whenever you have an inhibitor present, the curve really tends to get more sigmoidal the more that you have an inhibitor present. So, definitely has an effect, and is a good feedback controller. All right, step 4. This is where things get exciting. Here, we're actually going to take our 6, remember we've had 6 carbons all the way through so far. Here's where we're going to split it up. We're going to take fructose 1,6 bisphosphate and we're going to break it up into 2 3-carbon fragments. And this there's an organic reaction that this mirrors. It's called a reverse aldol condensation. I know when I taught organic chemistry, we didn't get to this. So, and I'm guessing Rachel, you, and Sarah probably didn't get to it either. So, it's not really that important for you guys to know what an aldol condensation or reverse aldol condensation is, but that's what it is. It's catalyzed by an enzyme called aldolase, and again, you can probably figure out from the name, aldolases, they catalyze either aldol or reverse aldol condensations. All right. I'm not worried about you guys knowing the mechanism of an aldol condensation. That's not important. What you do need to know is that the fructose 1,6-bisphosphate will split into 2,3 carbon fragments. All right. Remember these numbers. Carbons 1, 2, and 3 will directly form a molecule called dihydroxyacetone phosphate. And you can see the structure of it there. Carbons 4, 5, and 6 will form something called D-glyceraldehyde 3-phosphate. These are both sugar derivatives. They're derivatives of 3 carbon sugars. All right. It's an equilibrium reaction. I forget what the delta G is, but it's not very high. The delta G at biochemical conditions is negative 0.23 kilojoules per mole. So very, very close to zero, so it is reversible. Where we really want to be is at glyceraldehyde 3-phosphate, right? You had those two arms or two legs of this chart, right? That are each based on glyceraldehyde 3-phosphate. We made one of them, so one of them is ready to go. One of them, though, is dihydroxyacetone phosphate. So we have one more step designed to get both of these 3 carbon fragments in the place they need to be. So step 5 is going to be conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And if you look closely at the structures of both of these, go through and count the carbons and phosphorus and oxygens and hydrogens in each one of these. You'll see that they're the same, right? Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they're isomers of each other. So, to do this, what kind of enzyme are we going to need? We're going to need an isomerase. Boom. There we go. Specifically, this is catalyzed by something called triose phosphate isomerase. Again, the name tells you that it's going to rearrange the structure specifically of a triose phosphate. And we said these were both phosphate derivatives of simple sugars, so that works. And here you can see this reaction. It's got a small delta G, about positive 2.4. So it is also reversible. But at the end of step 5, you're ready to start phase 2. You got 2 molecules of glyceraldehyde 3-phosphate. Now we got to get them all the way to pyruvate. And oh my goodness, I'm getting wistful here. Remember, I taught this a long time ago. So these are 2 of my dogs I had in the past. They're sadly no longer with me. The one on top is Cricket, and the one on bottom is Davy. They were awesome dogs. So here they are taking a nap to illustrate the fact that take a deep breath, breathe in, take a breather before we get to phase 2. And that's what I'm going to do. I'm going to take a breather. We're going to make this the end of video 1, and in video 2 I will pick up where we left off and we'll talk about phase 2 of glycolysis. Alright, until then, have a good one. Talk to you later. Bye-bye.